Alors, bonjour tout le monde. Très, très content de vous voir en si grand nombre aujourd'hui. Bonjour à l'équipe du Chêne, vous êtes bien représentés. Et donc, aujourd'hui, en fait, je voulais vous introduire celle qui va présenter notre invité.
C'est comme Inception.
Oui, non, tout à fait. Je vais vous expliquer un peu le contexte de tout ça. En fait, il faut savoir qu'il y a maintenant presque 5 ans, Caroline Duchesne, donc, qui est chercheur ici à l'Université Laval, qui est dans le même département que moi en microbiologie et informatique, mais qui a ses laboratoires à l'UCPQ, a eu une illumination et a décidé de vouloir organiser un gros projet collaboratif entre de nombreux chercheurs au Canada, mais ailleurs aussi, sur l'étude des gènes de résistance aux antibiotiques dans l'air, dans les bioaérosols. Et donc, ce projet-là a été financé par le programme Frontiers du CSNG, une subvention de 4 millions pour 4 ans. Et ça a permis à Caroline de pouvoir mettre en place plein de choses, dont, entre autres, la conférence que vous avez eu la chance, pour ceux qui étaient là, de voir de Florent Rossi cet hiver, qui a parlé de la microbiologie et des gènes de résistance aux antibiotiques dans les nuages. Et donc, ça fait partie de tous les projets super flyés que Caroline a réussi à combiner ensemble sur l'étude de la résistance aux antibiotiques. Très heureux avec mon équipe de participer à ce projet-là via les projets de doctorat et de maîtrise d'Alicia et de Sarah. Je ne me suis pas trompé. Et puis, donc, tout ça pour dire que c'est un projet qui a pris une tournure super intéressante au cours des 4 dernières années. Et Caroline m'a dit que ce serait super intéressant de pouvoir accueillir le conférencier qu'on va avoir aujourd'hui dans ce contexte-là. Et on trouvait qu'ici était un bel endroit pour permettre cette conférence. Et donc, je vais inviter Caroline à venir nous parler.
Merci. Bon, ben, bonjour tout le monde. Je vous dis aussi bienvenue aujourd'hui à la conférence. Je vais juste terminer ça. Bon. Alors, on est vraiment heureux d'accueillir Kim qui a passé les 3 dernières semaines avec nous dans le labo, entre autres parce qu'il voulait se familiariser avec les méthodes d'étude des bioaérosols, plus particulièrement dans le contexte d'antibiorésistance, mais aussi en général au niveau des méthodes d'échantillonnage, de traitement d'échantillons, puis aussi d'analyse par la suite. On s'est rencontrés pendant la COVID de façon virtuelle. King voulait venir faire un postdoc dans mon labo, de chose à l'autre. Ça, c'est pas révélé faisable. Cependant, lorsqu'on a fait notre mini-symposium à l'automne dernier, on l'avait invité comme conférencier parce que c'est un jeune chercheur qui s'intéresse à l'antibiorésistance, dans l'environnement aussi, puis bien sûr, qui veut développer l'aspect des aérosols. Il a donné la conférence d'ouverture de notre symposium, malheureusement en Zoom, parce que les délais au niveau des visas, ça avait été trop compliqué. Alors, on s'était dit, ce n'est que partie remise, et voilà, il est venu nous trouver pendant. Il a passé, d'ailleurs, je tiens à remercier les membres de mon équipe pendant les dernières semaines qui se sont vraiment bien occupés, qui l'ont mis sur le terrain, qui ont montré des échantillonneurs, qui ont montré plein de choses qu'on faisait. Ça a permis beaucoup d'échanges, qui, je l'espère, on va rendre son voyage fructueux. Alors, sans plus tarder, je vous le présente. Il a obtenu son doctorat de Technologia, donc c'est pas un PhD, c'est un doctorat plus technique au niveau de l'analyse de l'eau en 2016 à l'université de Chuan, à Pretoria, donc en Afrique du Sud, mais il est originaire du Cameroun au départ, donc il a fait son bac sa maîtrise au Cameroun. Et maintenant, il est professeur associé à Applied Environmental Microbiology, Antimicrobial Research Unit, College of Health Sciences à l'Université KwaZulu-Natal à Durban, en Afrique du Sud. Donc, il quitte tout à l'heure, il va aller faire une conférence à Saint-Hyacinthe dans le groupe auquel Steve et moi, d'ailleurs, on fait partie, le CRIPA. Donc, il va aller parler un petit peu de ses intérêts de recherche. Puis on est super heureux de l'accueillir aujourd'hui.
Merci beaucoup, Caroline. Est-ce que je suis audible? Ah, OK. Écho? C'est bon? Ah, OK. Je ne suis pas habitué au micro. J'ai souvent peur des caméras, donc étant devant l'écran, là, OK. Mon groupe de recherche s'appelle Antimicrobial Research Unit, donc je vais aller du français à l'anglais directement parce que je suis plus confortable avec l'anglais quand il faut entrer dans le sujet de la microbiologie. Bon. Antimicrobial Research Unit. La directrice, c'est le professeur Sabia Isaac. Elle va être ici, peut-être elle est déjà au Canada même. Elle va être à Montréal la semaine prochaine. Elle est l'une des keynote speakers de EDA 7. Donc, ceux qui vont être à EDA vont pouvoir la rencontrer. Caroline, je vais te la présenter. Et je suis avec ce groupe depuis 2017. Lorsque le groupe a commencé en 2013, mais quand j'ai rejoint le groupe en 2017, il n'avait personne en environnement. C'est comme ça que je me suis retrouvé là-bas. Je faisais euh, les humains, on dit les humains, n'est-ce pas? Les hommes et les animaux. Et je suis venu donc pour l'environnement. Aujourd'hui, je vais présenter sur la résistance antibiotique, une approche One Health. Et je vais plus parler de ce qu'on fait dans notre laboratoire. Et donc, ce que je vais présenter vont être des résultats de nos recherches et certains projets que j'ai faits. Il y a peut-être un ou deux projets que j'ai faits lors de mon PhD. So, let me now come back to English. When you look at this picture, what comes to your mind? How many components do you see there if we are talking about a One Health? You will quickly realize that we have animals, we have the environment, and we have humans. That is a complex picture, as simple as it looks. And this picture was taken during my PhD. I was actually sampling that river. So, in order to go into the One Health concept, let us first of all try to put antimicrobial resistance in the context of pandemics. When we look at pandemics throughout the world that have hit the world, and if we focus just on the 20th century and the 21st century, then we have had three major pandemics. We have had the Spanish flu, we've had the HIV/AIDS, which is still going on, and we had the COVID. When we look at the number of people that died out of these pandemics, we start to understand why we call them that way. But why should we place AMR in the context of pandemics? So those are the three we are talking about. So if we bring AMR in this context, let us look at the number of people that died in 1 year, that is in 2019, just because of either directly linked to antimicrobial resistance infection or infections that had an association with AMR. We are talking about close to 5 million people in a year. Now, if we also look into statistics, when Jim O'Neill was called to make a report, an economist was called to make a report on the level at which AMR was affecting the globe, and he projected that if nothing is done, then by 2050, we are talking about 25 years to come, by 2050, we will be losing 10 million people every single year. Now, if we take 10 million people every single year and we locate it in the history of pandemics, then we are talking about 5 years for AMR to surpass every single pandemic that has existed. That is scary. And the researchers who are in the hall with me will agree that getting research funding these days is very, very difficult. And yet, AMR is costing the world over $100 trillion in global economy, meaning that we are going down and down and down when the funds are concerned. So the organizations out there, the World Health Organization, the Food and Agriculture Organization, or the big organizations, and including the United Nations that had to bring in AMR as one of the global health issues. I think that was the 5th health issue that has ever entered into the UN National Council. So they talked about this and they came out with this nice concept, which is the One Health approach. And this One Health approach simply says that the only way we can succeed is if we come together as one. To give this as a simple analogy, I want to take a story of an old man back home. I don't know him anyway. I was told the story, so don't ask me who the guy is. So he called his children and he gave each of them a broomstick, small stick for them, like the ones we use to make our baguettes. And he told them to break them. And each of the children could break the stick. So it got broken easily. But then he took a bunch of those sticks and he gave them, each of them, and he said, break them. All of them tried and they couldn't break it, no matter how hard they tried. Simply meaning that the One Health concept says if the environmental microbiologist is going to work alone, if the veterinary microbiologist is going to work alone, if the clinical microbiologist is going to work alone, I'm saying microbiologist, but you can put it in science completely, then there is no way we are going to defeat AMR. So we need that complete communication, right? We need that tight communication, because if we are tight in that communication, then we are like that bunch of broomsticks that can't be broken. But if we stay individually, then we run the risk of falling to AMR in the years to come. We might be talking about 2050. In some other countries, by 2030, that's just 6 years to come, it's already going to be rough. So what, therefore, we decided to do at the Antimicrobial Research Unit, most times I have presented small portions of our work. Now I'm bringing in some sort of a global picture to what we are doing. So what we decided to do was therefore to look comprehensively on antimicrobial resistance within our setting using the One Health approach. And we had different work packages in our attempts. We had to engage in surveillance, look at the resistance mechanisms, how this resistance is disseminated, and are these organisms even pathogenic, and can we inform policy on what to do? Because antimicrobial resistance, as in Canada, I am sure, in South Africa, is part of the the National Action Plan. And it's been pushed from 20 to up to 2024 now. It's gone to 2030. As the years are going, it keeps on coming in, which means that it's a really important aspect. So how, therefore, do we conceptualize this one? When we look at our conceptual framework, we say we want to do surveillance. This surveillance will be in humans. The surveillance will be in animals. The surveillance will be in the environment. And what is the ultimate goal is that we should be able to understand what is the level of resistance that we have, to which antibiotics we are having resistance, and what is the overall burden. Because when we have that overall burden, then we can come in with our interventions. And these interventions can be biomedical, we can have clinical or veterinary. We can go right down to social and behavioral, because we know this is another aspect that comes in. And we can talk about discovery of antibiotics, of different antibiotics, and we can look at interventions at the level of the policy. Again, the overall goal is that we need to cover all these aspects if we need to address AMR. Our works mostly focuses in this district. Don't ask me to pronounce it. I've never been able to pronounce it. If you can pronounce it, you raise your hand, I'll give you the mic and you do that. I don't know that place we studied in that district, that district, right? So we took this district because it has most of the primary healthcare facilities that we have. It has district hospitals and regional and tertiary hospitals. It's also a mix of urban and rural mix. And we also have in this same district intensive poultry and pig agriculture. And the pig and chicken litter that is produced in that place is used to fertilize the farms still in the same place. And it has 6 wastewater treatment plants, most of which treat their water and send it back directly into the river. So you can see why we choose that. We kind of have a picture of what we want to do in this place. Obviously, we got all our necessary documents to ensure that we carry out our work within the norms. And we also ensure that every participant, especially on the human side, agreed to carry out the work that we wanted to do or to participate. For reasons which we will understand, anonymity is one thing that is key when we are carrying our research. So it's very key in this place. We are not supposed to divulge the names of our participants. And at times, we are not supposed to mention the names of the wastewater treatment plants, because South Africa is quite big on its wastewater treatment plants monitoring. So if I call the name of a wastewater treatment plant, I might go back and lose my job. What is our methodology? With the methodology, we use the GLASS protocol, which is the Global Antimicrobial Resistance Surveillance System for the humans, and we use the AGISA protocol for the animals. For the environments, you would agree with me that we don't have such a protocol that tells us what we need to do with the environment. The environment is so complex that everybody dives in there and learns how to beat the swimming strokes as they go ahead. So we kind of developed our own approaches to work based on existing literature. So when we look at water, we were sampling water every 2 months, twice a month. We sampled for over 7 months. And then we were sampling the influence of wastewater treatment plants, the effluent, upstream and downstream of those wastewater treatment plants, because those wastewater treatment plants service the whole that environment. So they also get wastewater from domestic, they get domestic wastewater and agricultural wastewater. And for soil in this area that were fertilized with chicken litter, we started sampling the soil before the chicken litter was applied, and we sampled after the chicken litter was applied. We went ahead to do phenotypic antimicrobial resistance using the diffusion method where possible, and in some cases, we were using the Vitek 2 when we had a lot of isolates, especially when we wanted to compare with clinical isolates. For the genotypic ones, which you are very conversant with, please don't ask me questions on any genomic thing that you see there. I might not be able to answer that. You are experts in that, I am not. There we did whole genome sequencing, and we wanted to see the overall resistome and at times to understand the genetic environment of the organism. For example, if we saw a resistant gene in an organism, was this carried on the same gene fragment as a mobile genetic element? Was it carried on a plasmid? Is there a possibility of transmissibility of this gene? Because if we have a resistant gene, it just doesn't mean that it's a problem. We needed to know if there's fluidity in this gene to go. So we had to go into whole genome sequencing to understand all these aspects. On the surveillance in the human sector, what did we find? If you look at the total there, which is in blue, and active sentinel surveillance, what had the active surveillance is that we looked at one hospital that was representative of the whole area, and we say every sample was coming there so we can analyze from there. Then we also had cases where we went into individual hospitals and collected samples. Then we had cases where we just collected any sample that came, whether it was from a patient or not, any sample that came from the hospital. The one thing that we realized here is that what different levels of resistance that were present in our samples. So if we look at this, then if we look at it carefully, then we consider the comprehensive surveillance, right? The comprehensive surveillance, which is the green, we might say that we are overestimating our antimicrobial resistance. But if we do not do that, if we go with the passive, it means we are underestimating. So the question now is, if we are underestimating by monitoring continuously, and we are underestimating by just collecting at one point and analyzing, what then is the good method to use? Then we go to the animals, and in the animals, we go to the poultry. This one was done by one of our master students, Stephanie Pile. She's now in the Netherlands. She just completed her PhD in bioinformatics. So she was the one who did the study during her work. The one thing that we see here is we also had different levels of resistance. We use the farm to fork approach, which means that we took the chicks from when they hatched until when they were packaged for the market. So we were using that farm to fork approach. So we sampled weekly in the chicken farm until the chickens went to the abattoir. We followed them to the abattoir. Then we collected samples even that were packaged. What is interesting here is that isolates, when we did pulse field gel electrophoresis on this isolate, we saw that isolates from the various sectors, that's the retail, the water that was coming from the farm, and some of the litter clustered together, which means that there was some sort of a fluidity moving from the farm right up to the abattoir, which again brings the possibility that we might have had it on our table already. Then we also looked at other organisms. We did the same with Enterococcus. We did the same with Escherichia coli, Salmonella, and Staphylococcus aureus, and we got almost the same output. We did the same now in the pigs, and it was still similar output that we were getting. When we go to the environment, this one was part of the works that I did for my PhD work. In this case, I was looking at river water that was impacted by 4 different wastewater treatment plants. It was impacted by agriculture. And the picture that I showed you at the beginning was part of this project. In this case, I was looking at the river water column and the bed sediments. We will understand that most times when we say we analyze the rivers, we mostly collect the water column and we analyze. But the riverbed sediments, it has been proven that it will contain more organisms than the water column. So in case of disturbances, I published another paper on that. In case of disturbances, we saw that the risk of infection increased from maybe 50, 70 to 100% if the riverbed sediment was disturbed. So, and also we realized that these organisms could survive in riverbed sediments longer than they could survive in the water. But what is interesting here is that we had resistance to different antibiotics in their water and in the sediments. In this other work, yeah, we looked at extended spectrum beta-lactamases in the Umgungu something district. That one. Yes, in that district. And here we did a point prevalence of 6 wastewater treatment plants. We wanted to see if we get out just randomly and sample those wastewater treatment plants on a single day. What do we find? Do we get a hit or do we get a miss? And when we went there, we found out that we had antibiotic resistance E. coli at different points. And when we went on to do real-time PCR, we found out different beta-lactamase genes. These are just for the genes. They had resistance to other antibiotics, which I'm not just putting. One other interesting thing is that we didn't understand some of these things, right? So at times you may have to ask questions in some places that we don't know. And I brought in this one because I had to go consult with my ancestors to ask them what was happening. And this work that we did, we went into a cemetery and decided to sample water that was flowing inside the cemetery. We sampled soil inside the cemetery because we estimated that there would be little human impact in the cemetery. It is closed area and everything. And we did surface water, the surface soil, and we did sampling at 2 meters, that's 6 feet, where we go when we are normal breeding. And we found different resistances at different points. We also decided to do 16S sequencing of the samples that we found in that same area to see if we could, maybe what we were picking were just growing, but what could we see with the 16S? By then, we didn't have the means to do shotgun. If I still had the samples, I would have tried that. What we realized is that we had different signatures. When we did the functional metagenomics on our samples, we had different signatures. But one thing that we need to identify here is that although at a smaller scale, we found at the surface and below 2 meters, we found antibiotic resistant gene, antibiotic resistance to beta-lactamase. We found signatures associated to that. So that therefore brings us to our whole aspect of the One Health. My video has decided not to play, but it was a nice video which I wanted us to see. I hope it doesn't freeze my screen. Okay, it has refused to play. But one thing that this, there we go. It refused to play. One thing that you would have seen is that these 3 sticks standing on the ground stand the way they are. But interestingly, if we could see it, this man succeeds in placing the 3 sticks. I don't know how, but he places them such that they stand, and he can climb and stand on them without tying anything. Which brings in that concept of the One Health, saying that individual sticks will fall, but the sticks put together in the proper manner, which is proper communication between the 3 fields, would support the weight of him. So therefore, when we look at the human and environment interface. This one was done by our PhD student. He's now a postdoc with us for the second year. And he looked at E. coli in wastewater treatment plants and compared them with clinical isolates, which we extracted from databases. What we find here is that we had down here, for example, we had a clustering of our environmental samples with human samples, the same way in the other side, and we also had animal samples clustering with environmental sample, which gives a certain level of fluidity of resistance between these two sectors. This was after whole genome sequencing. When we look at human-animal interface, this was also done by another student, Daniel. He's now based here in Canada. And he was looking more at S. aureus, at Staphylococci in Staphylococcus aureus. And what we found is that most of our isolates, or all our isolates, were of the ST8, but these isolates, again, yeah, there we go. So those are our isolates clustered with some other isolates in the USA and some other isolates still within South Africa. Again, saying that although we were finding our isolates in South Africa, whole genome sequencing and comparative genomics showed that these isolates were related to other isolates that had been isolated in humans and in animals in other sectors. And these were isolated from poultry in South Africa. We look again animal environments. This one was done by our student, Dorcas, who had done the work in the field farm where we had collected chicken manure and we had applied manure and then collected samples before and after. In this case, Dorcas also saw that here we had poultry samples that were clustering, that's poultry litter that was clustering with the amended soil, which means that the antimicrobial resistance had moved from the chicken litter and had moved into the soil. Remember, this chicken litter came from the same farms in which we did the other studies. So we are still within our same district. And we had 2 isolates of the chicken litter that clustered just by themselves, meaning that the chicken litter was bringing something unique that was not present in the environment. And we have the environment and the soil. And the same applies on all these other aspects where we had isolates that were clustering based on their genomic characteristics. So what then do we bring out of this? We want to understand that based on this analysis, we see that antimicrobial resistance, we have antimicrobial resistance in each of these sectors. That's in the human, the animals, and the environment, and that there is transmission between and within and between these sectors. These isolates carry mobile genetic elements, which means that they have the possibility of transmitting the resistant genes that they carry. And not just the genes are present, like in the environment, for example, we also have the bacteria themselves that are present. So what we know, therefore, is that AMR can evolve and it can be transmitted within these sectors. And each component serves as a reservoir and a means of transmission. And that these drug-resistant bacteria are very fluid. What we do not know is how and to what extent are these various sectors contributing to AMR. If we were to quantify AMR into general, are we able to say the human sector contributes more than the animal sector, more than the environment? When we look at the use of antimicrobials, for example, we will realize that while up in the West, for example, more antibiotics will be used for humans, in down south, we get more antibiotics used for agriculture. So how then do we quantify? Is it possible to do that? What is the exact role of AMR, of the environment in the transmission of AMR? Do the bacteria come into the environment already resistant, or do they acquire resistance in that environment? And if they acquire resistance or they come inside the environment already resistant, how then, what is the role of the environment in transmitting this to humans and in transmitting this to the to the animals, and what is the impact itself on the environment. So there is all this complexity that is there, and what is the overall human and animal health implication? Up till date, we are still struggling on QMRA. If we find this resistance with other isolates or virulence organisms, for example, we can calculate the exposure risk and the risk of infection which somebody will get if they expose themselves to virulent organism. But the question is, can we do the same calculation for AMR? What are the chances that somebody who picks up a resistant bug is going to develop an infection that is linked to resistance? Those are questions that we have to answer. So the challenge, therefore, is that surveillance is taken in silos. We work in our different groups, humans, animals, environments. At times, integration of these three components is usually complex because of the difference in the matrices. And genomic studies have shown that although we may have similar strains, similar genes, and mobile genetic elements in different One Health, but yet we need to establish their actual dynamics and the risk associated with AMR transmission. So we therefore say that standardized methods for health surveillance protocols are needed. We need more sampling. With the cutting down of costs, it becomes challenging to get more sampling. I was discussing that with Steve today, and I thought that the problem was just down south getting funds, but it's quite challenging in this side too. What is the sampling frequency? I presented work and I said we did a one shot. How often do we need to go to establish that? If we say as many times as possible, then we still go back the point of, do we have the funds to do that? Do we have standardized protocols? Do we have established SOPs that can be circulated and applied to every single setting? And when we think about that, the question is, so what? What are the transmission dynamics that we get there? What are the risks? And so from this, we have learned that if we want to do active surveillance, which is important, it's very expensive. But again, not only is it expensive, but it introduces a bias because we are only looking at specific organisms. We are only looking at pathogens, for example, in humans, and we are looking at commensals in animals. We are not looking at pathogens. So there is a bias in comparison. So we decided to look at wastewater-based epidemiology because the idea now is, instead of going for active surveillance where we have to carry a massive sampling, we drew inspiration from these other studies that were conducted in Sweden. I'm sure you probably know about this work, the global surveillance, that involved about 100 countries. But one thing that comes out here again is that low and middle income countries had the highest burden of AMR. And when we also look at urine samples in primary healthcare patients, again, still in Gothenburg, and we see that there was quite a good correlation between the isolates that were gotten from patients and wastewater. The same applied for hospitalized patients still there in the other case in 10 European countries. So if we also look at the massive surveillance that took place with the COVID, I'm sure you people also did a lot of sequencing this side. What we found out with these works is that the cases of COVID that were reported were matching with those that were reported in wastewater, which makes wastewater-based epidemiology a good approach. So in our research group, therefore, we said, if we can't do active surveillance, what should we do? And we came up with these beautiful alarms to raise an alarm saying that we are looking at alternative antimicrobial resistance monitoring system. And what do we intend doing in these alarms is that we have two hypotheses. And the first hypothesis here is that we are going to look at informal settlements. We are going to look at vulnerable people because we think that these people are more prone to infection and yet they are not considered. Unlike here, where everything is a little bit structured, South Africa has a lot of informal settlements that are like this, where they don't have any service delivery, they don't have any water, so they use the nearby bushes, they use the nearby streams as their point of discharge. So studying the dynamics of antimicrobial resistance there becomes interesting. And we want to understand what, therefore, if in the hospital we can tell the resistance of the people, how are we going to tell this? Can we get a proxy? Can we get the river that flows across this community to tell us what is in the community? And we have a high rate of HIV on the same areas. The second hypothesis is that, can we do single cell genomics on our isolates and get appropriate results? If we do that, then we are going to base more on culturable, right? But if we go with metagenomics, will metagenomics give us a better representation of the analysis and give us a better picture of what we want to see? Because in this case, we are picking viable but non-culturable bacteria as well. We are picking everything that is present in our metagenome. We are picking gram-positives, which most time we ignore. We might be picking as well environmental DNA, which is not found in any of the bacteria. So our research question, therefore, comes into say, is the sewage, like for the COVID, like for the urinary tract infections that we saw, like for the COVID, will sewage surveillance or surface water be a good proxy to conventional monitoring methods? Will metagenomics give us a better picture to identify or to quickly inform of new resistant threats in these vulnerable communities? And do we have distinct genomic signatures that we find in AMR using metagenomics compared to other approaches if we look at our subpopulations? And what are the societal and behavioral patterns? Because one other problem that we have, I don't know if you have them here, is that antibiotic use is a complex process in those type of communities. They go to the hospital and they are sick and the doctor tells them you have a flu and they say, no, but I need antibiotics. And you have to give them when they insist. Now, do we have these behavioral patterns also affecting AMR in this place? We have the black market, which is not talked about. The government may control all the resistance that we find, but can we control that in the black market? Those are all behavioral patterns. How many times do we have antibiotics that are expired in the house because it worked for one person and we have to give to the next person? It's very common. So we are also looking at that. So will alarm give, and can we put all this in some sort of a mathematical model using the genomics approaches, using the surveillance that we have, the cultures and everything? Can we try to model this and say, this is what will happen with resistance within a certain period if we have all these factors put together? And if the approach we are proposing is going to be a concept that can be scaled, why not globally? Those are the various work packages that are involved there. You can just see it's almost similar. We will have to do so many things to achieve those goals. There are all 10 of them. And what we envisage with this is that we are able to establish an alternative monitoring approach for AMR that will help give an early warning in the case of an outbreak, to prevent outbreaks. We hope that we are going to contribute to the whole global repository of bacterial whole genome sequencing by the works that we are doing. Currently, we are receiving from 6 countries isolates that are coming from different sites that are still linked to this, and we are already at over 1,500 isolates, and we are going to be receiving these isolates until the end of the year. So we are looking into sequencing a very huge bank of isolates. And then we hope that we will generate enough potentials to continue when we retire. I still have a long way, so don't ask me if I'm going to be among them. And we also think that this is going to strengthen the health system because we will be able to predict some of these antimicrobial resistance threats before they even come in, and will enhance capacity in AMR. These are the people who give us money. They are cutting the money drastically. And in all these that I have presented, the question is, where are the bioaerosols? We have a gap, and we have identified some of them, but there is more to be identified. And with that, I will say thank you for listening.